Good afternoon, Dr. Tolliver, and whoever may be watching this presentation. My name is Quincy Hall. I'm a senior biology pre-med major from Chicago, Illinois, and I chose to do my presentation on the structure of the coronavirus, specifically COVID-19, and how we can use the information from the structure to create new vaccines in the future. So, in order to understand the structure of COVID-19, we must look at and do some background on what a coronavirus is in general. Now, coronaviruses are a large family of viruses that contain cause respiratory illnesses, ranging from the common cold to more severe diseases such as MERS and what is spreading today, which is known as COVID-19. Now, COVID-19 is a new disease that we've never seen before. It's unknown in humans. We've never seen it before until recently in Wuhan, China. An outbreak began around December 2019. Now, the CO in COVID, just for a little background on the scientific term COVID-19, the CO in COVID stands for corona, the VI stands for virus, and the D stands for disease. And the 19 comes from the year in which the virus was first discovered. So, like any other virus, the novel coronavirus is a germ that tries to burrow into the cell and turn it into a virus replicating factory, so to speak. If it succeeds, it can produce an infection, in this case, a respiratory disease. So the type of cells a virus targets and how it enters them depends on the way the virus was built. Now, this virus gets its name from a series of telltale spikes. It's like tens or even hundreds of spikes that circle its blob-like core and form a crown or a corona, hence the name coronavirus. Now, very recently, innovations have begun in imaging techniques and have enabled researchers to peer closely at the novel virus's spikes, so that so much that they've created a model of one right down to the atoms, and are beginning to reveal everything about the structure of the virus. Now, because the coronavirus is not alive, it makes it so much harder to kill the virus. Mainly because it has a very, very clever disguise. There are sugars on the virus that dot the outside of the spike, and it's very similarly to just how sugars dot the outside of regular human cells, and it's almost acting as a camouflage. Now, this carbohydrate camouflage is it makes the virus more difficult to be detected by the human immune system, because it's almost similarly disguising itself as a regular human cell. So the immune system doesn't know what is toxic and what's not. It doesn't know what needs to be removed and what's not. So through research, we have seen that the coronavirus seems to have a mechanism where it attaches to cells by opening and closing. Each spike is made of, we've seen that each spike is made of 3 identical proteins, that are twisted together. And a group of scientists have captured images of these proteins opening in the spike's cap-like apex before and during the attempt to bind to a receptor. Now, studies on SARS and MERS causing viruses indicate that all 3 proteins in a spike have to open for it to gain access to a cell. Now, there is hope that a vaccine may be able to exploit these openings. Experts say that we are like a year or so away from vaccines, it seems like, but if it is possible that we can trigger antibodies that strike areas of the protein that are exposed when it opens, we'll be able to counteract the lock and key mechanism and use that as an advantage to take down the virus. Now, the ideal vaccine would trigger antibodies that attack the spike stem. which is so similar to the coronaviruses in general that a single vaccine might protect against several strains instead of just one. Um, One treatment might be to block receptors so the spikes have nowhere to attach, which is sort of different from just um, countering the fact that we're essentially, we know that the virus is going to have the lock and key mechanism and it has to open to attach. But instead of having it attach in the first place, we just block the spikes so it's not able to attach at all. Now, um, they're not close to a vaccine, but there are many important, there are even more important discoveries that have been made on the structure of the virus that allow us to still stay safe and still stay protected, even though we don't fully know the extent of the coronavirus and how long it takes to how long the virus lasts on surfaces and things of that nature. So um, this is why the CDC and everywhere you see people preaching to wash your hands and stay clean and Lysol. This is because about 20 seconds of contact with soap and water, the the fragile fatty part of the membrane that holds the virus together breaks down. Disinfecting agents that have more than, uh, 60% alcohol puncture and destroy the virus in a similar way. So basically, hand sanitizer with more than 60% alcohol will have a similar effect to breaking down the fatty acids that hold the membranes of the virus together. Now, extreme heat is also a way um, that we can counteract the coronavirus. Almost heat hot enough that it's near boiling point causes the protein spikes to unravel and lose their shape, and it essentially deactivates them, which makes them not uh, properly operate like how they were if they weren't under extreme heat conditions. Now, one thing that is not true is that a fever is not hot enough to do this. Now, some people have been thinking, um, it's been reported that some humans have thought that having a fever means you don't have the coronavirus because it's hot enough to kill it. But a fever is not hot enough to unravel the spikes. It's not that hot, and it isn't near boiling point. Um, so it won't be that hot to affect the spikes and have the coronavirus lose its shape. Lastly, ultraviolet light has been used as a disinfectant in many hospitals for the last 100 years or so, and also in water supplies. Basically, it shatters the genetic material inside the virus, bacteria, and other microbes. So, but it doesn't always work uniformly, and it wouldn't be able to use with people around because it damages the healthy human cells that we need. So it'd be counterproductive to use it as a coronavirus agent to stop the coronavirus. So my name is Quincy Hall, and that was my presentation on the coronavirus. I feel like we can make further findings and implicate new vaccines and procedures and prevention measures based upon the structure of the virus. Now that we know that it's a lock and key mechanism, if it's possible to block the spikes, I think that is a better alternative rather than using the lock and key mechanism to our advantage. I think that's a better alternative. And we just have to wait and see if scientists can figure out a way to put that procedure into place and get it to a point where we can extract a stable vaccine for the virus. My name is Quincy, and thank you for the time on this presentation.